In the next section, we'll describe the physiology of reflexes. In this section, we'll discuss reflexes. There are several different types of reflexes that are embedded into the circuitry of our spinal cord, and they control contraction of muscle without our having to consciously control that type of contraction. The first type of reflex that we'll talk about is going to be the deep tendon reflex. The deep tendon reflex is mediated through what's called the muscle spindle. The muscle spindle is shown here on the right side of this figure. It is the football-shaped organ within the muscle that's shown on the right. The muscle spindle is made up of modified muscle fibers. These are also known as intrafusal fibers. This is to contrast these fibers with the traditional regular muscle fibers known as extrafusal fibers. So these intrafusal fibers are arranged into these football-shaped organs known as spindles, and these spindles serve as stretch receptors. When the spindle is stretched, the innervation of these spindles gets a signal. These signals travel back into the spinal cord to tell the spinal cord and ultimately the brain that the muscle has been stretched. The afferent innervation of these spindles, that is, the innervation that carries this information back to the spinal cord, is carried via type 1A and type 2 sensory neurons. There is also motor innervation to the spindle. The motor innervation is carried by what are called gamma motor neurons. Also shown in this figure in green are the alpha motor neurons, and recall the alpha motor neurons are the neurons that innervate the extrafusal or the contractile elements of muscle. The next slide illustrates a bit more about spindle physiology and illustrates why spindles need to have gamma motor innervation. So the top figure shows the normal parallel organization of extrafusal and intrafusal fibers. Extrafusal shown on the left, intrafusal shown on the right. The top right tracing with a series of vertical lines represents the action potential activity in the afferent innervation of that spindle carried by the type 1A or type 2 sensory neurons. And one sees that with no stretch at all, that there is a particular spontaneous rate of firing of action potential production by those sensory neurons. When the extrafusal fiber is stretched, the spindle, because it's arranged in parallel, is also stretched, and one can observe that on the right middle panel that the action potential firing rate increases substantially as that spindle is stretched, thus efficiently coating the stretch in that muscle. But when the muscle is contracted, and if the spindle itself is not also simultaneously contracted, one can observe that the spindle is no longer able to adequately encode the length of that muscle. One problem is that the so-called slack in the line in the connection between the spindle and the rest of the muscle itself is too great in order to allow this spindle to do any kind of efficient coding of a length in the muscle. And therefore, even though the muscle is contracting, the spontaneous rate of firing of that spindle as encoded in that type 1A and type 2 fiber is really no different than it was in the case on the top when there was no change at all in the length of the muscle. Therefore, there needs to be a way to adjust the length of the spindle to match the length of the muscle whenever the muscle contracts. For that reason, spindles are innervated by motor neurons known as gamma motor neurons, such that there is now symmetry in this innervation. There is alpha motor neuron innervation of the extrafusal fibers and gamma motor neuron innervation of the intrafusal fibers, and this simultaneous activation ensures that intrafusal fibers always maintain a similar length as the extrafusal fibers such that they could encode changes in length much more efficiently. The organization of this is shown at the bottom of the figure. On the right, one observes the lateral cortical spinal tract from the brain, which carries the efferent signals from the brain down to the spinal cord itself. The lateral cortical spinal tract innervates the alpha motor neuron, shown in green, and in addition to that, it also innervates the gamma motor neuron, thus establishing a mechanism by which simultaneous or parallel activation of these two muscle groups can occur. In addition, shown on this figure is the dorsal root entry of the type 1A or type 2 sensory neuron, which carries afferent innervation from the spindle. And as shown here, this afferent innervation will directly synapse in a monosynaptic way onto the alpha motor neuron, which will become more important in the next slide. So if we were to put this all together, we can observe how something like the deep tendon reflex works. The deep tendon reflex, as shown here, is being executed on the patellar tendon. So if there is stretch on the patellar tendon, the muscle spindle will stretch and will sense this and send this information back through the dorsal root. That afferent information will synapse in a monosynaptic way onto the alpha motor neuron, and that axon is shown in purple to cause contraction of the homonymous muscle, of the quadriceps femoris. In addition, a branch from that axon will synapse on an inhibitory interneuron to cause relaxation of the antagonist muscle, shown here as the hamstring, to allow relaxation and adequate extension of the knee in order to get that muscle back to its original length. This is a monosynaptic deep tendon reflex. One point about the biology of these intrafusal fibers is that there are two different types of intrafusal fibers. Remember, these fibers are made up from the fusion of multiple other cells, therefore they have multiple nuclei, and the nuclei can be arranged in different ways. They are nuclear bag fibers, which have all the nuclei bunched together in the middle, and nuclear chain fibers. Both of these fibers are found in muscle spindles. The majority of them are nuclear chain fibers, and there is some slight differences in the way that they are innervated. The type 1A afferents innervate both nuclear bag and nuclear chain fibers, whereas the type 2 afferents primarily only innervate the nuclear chain fibers. Both sets of fibers receive gamma innervation. One important clinical manifestation of the deep tendon reflex is that the deep tendon reflex is under a substantial amount of inhibitory control. And that's illustrated in this example of a reflex not from skeletal muscle but from smooth muscle, but the same rules apply. If one were to observe the situation on top, the normal bladder function, there is a reflex to sense the stretch of the bladder, and when the stretch achieves a certain level, to reflexively empty the bladder. Under normal circumstances, that reflex is suppressed. But under situations when the descending inputs from the brain are suppressed, typically via spinal cord injury, now this reflex is no longer suppressed, and one finds that with small changes in the volume of the bladder, that there is reflex emptying of the bladder. This results in incontinence and is a particular form of neurogenic incontinence known as a spastic bladder. But this is a general rule, and that is when the descending inhibition is removed, one will find hyperactive deep tendon reflexes. Another reflex of importance and can be observed clinically is the Golgi tendon reflex. The Golgi tendon organ is a stretch receptor that is found in the tendons of skeletal muscle and is arranged in series with the skeletal muscle fibers or with the contractile apparatus itself. The innervation of the Golgi tendon organ is shown here in purple. And interestingly, if one were to follow this purple axon along, one will see that when the Golgi tendon organ is activated, it sends its information along this afferent axon. That afferent is going to innervate an inhibitory interneuron shown in the spinal cord, also in purple, and that inhibitory interneuron will inhibit the alpha motor neuron that innervates that homonymous muscle. Therefore, when the Golgi tendon organ is activated, it will cause relaxation of the homonymous muscle. This is actually the exact opposite of the deep tendon reflex, and that actually is also shown in this figure. There is a spindle shown towards the middle. The blue fiber is the afferent innervation from the spindle, and for comparison's sake, one can observe how that axon is monosynaptically connected with the alpha motor neuron, such that when it is stretched, it would cause that muscle to contract. It's thought that the Golgi tendon organ, its role is to protect the muscle from damaging stress. One can observe the activation of the Golgi tendon reflex if one were to examine a patient who has upper motor neuron injury and spastic posturing of their upper extremity. What you'll find is that with pulling of the flexed upper extremity, it will be very difficult to cause the upper extremity to extend. But then after a certain amount of pulling is done, all of a sudden there will be a great loss of tension and the arm will open up quite easily. This is known as the clasp knife reflex and is thought to be related to the activation of Golgi tendon organs, which causes the relaxation of the homonymous muscle in order to protect it from fighting against overwhelming force. And the last reflex we'll talk about involves a bit more integration, is often called the cross extensor reflex, and it's a reflex that allows us to rapidly escape from a noxious stimulus while maintaining our balance. So in the case shown here, if somebody was walking along and stepped on a nail, that painful noxious stimulus will be sent to the spinal cord. There are projections to the alpha motor neurons of the homonymous or of the contralateral side to allow that leg to escape the noxious stimulus, and that would involve activation of the flexor muscles of the leg. So you would activate the hamstrings, for example, to flex the knee, and you would also activate the musculature to flex the hip to allow yourself to escape that noxious stimulus. However, that's not enough. One also has to maintain posture via the extension of the contralateral extremity, and that's done through the crossing fibers that are shown in the diagram of the spinal cord shown on the top right. These crossing fibers will cause the extensor muscles, so the quadricep muscles of the contralateral side, to activate to allow, and conversely will cause the flexor muscles of the knee to be inhibited in order to allow activation of the contralateral limb, to allow stability to occur while you escape the noxious stimulus. It's important to recognize that all of this circuitry is built into the spinal cord, and all of this reflexive movement occurs without our conscious control. This concludes our discussion of the physiology of reflexes.